malnutrition process begins with the transformation of agrobacteria. For this demo, we will be fortifying Arabidopsis with lysine.
Here we have a plasmid that contains the gene for lysine production, and here we have the wild-type bacteria. All of the bacteria and the plasmid containing the lysine production gene are then combined into these two tubes. One's a positive and the other one is a negative control.
To transform the bacteria, we must insert the gene for lysine resistance into the bacteria. This is done by using a heat shock to open the cell membrane.
Then transferred to the ice to close the plasmid into the bacteria. The transformed bacteria will then be placed in an incubator to recover with nutrient broth. To increase transformation efficiency is called electroporation. This is where the cell is shocked by electricity to transfer the plasmid into it.
This broth has been inoculated with the transformed bacteria and incubated overnight. The broth contains the same selective ampicillin as the plate. The next step is to transform the plant with the lysine production. This is done by using a syringe to inject the transformed agrobacteria through the stoma. The transformed bacteria is then plated and grown. Selection media is used. This works by killing bacteria that are not transformed with the lysine containing plasmid. This occurs because the plasmid also codes for antibiotic resistance, and the media contains an antibiotic, in this case ampicillin. As seen here, the positive plate has growth while the negative plate does not.
Irrigation system. Plants are placed in the hydrogen beads, and small amounts of water drip through the tiny spouts, which are embedded inside the cubes.
A very thin layer of concentrated nutrient water solution is in the bottom of the tube, thus limited water supplies can be used. The roots grow into the solution and the exposed parts of the roots absorb the needed oxygen. And this solution contains nitrogen and zinc to biofortify the crops with additional nutrients. Nutrient film techniques can be used in water-poor regions like Myanmar with nutrient deficiencies. They therefore optimize the nutrient growth and crop yield. Is an ebb and flow system. Plants are placed in the rocky substrate in trays, which then flood and drain periodically. Small amounts of water can be used since it is constantly recirculated and recycled.
This growing Brassica rapa is an example of passive hydroponics. The plants are situated on top of an absorbent mat and take water through capillary action. Passive hydroponics is a cheaper and easier way to use hydroponic system on a small scale.